Our today's topic of discussion is animal and human-based research on myopia. During the late 70s, animal models of myopia were developed in monkeys, chicks and tree shrews. Form deprivation by frosted occluders was found to produce axial elongation and myopia in the occluded eye. 10 years later, the model was expanded to include myopia produced by negative lenses and hyperopia produced by positive lenses, applied to the eyes of 7-day-old chicks. The models have been extended to a variety of mammals, see reference for review. It is generally accepted that the mechanism leading to changes in ocular elongation involves local retinal image processing, which sends a stop or go message to the sclera, regulating scleral matrix properties and its rate of elongation. Somehow the growth mechanisms are sensitive to where the principal image plane is falling. If the principal image plane falls behind the retina, the eye tends to grow more, and vice versa. When the image plane lies in front of the retina, the choroid thickens and the eye stops growing. Most studies on animals have been carried out during a period that would be equivalent to the first year or two of life in humans. In that period, non-treated congenital ptosis, corneal scars and non-treated dense congenital cataracts often produce axial myopia in humans, similarly as form deprivation develops in animal models. He signaling cascade from the retina to the sclera is still under study. Amacrine cells have been implicated in retinal processing of the signal. The choroid and the sclera show biochemical changes associated with ocular growth in the animal models. Two drugs which have potential to prevent myopic progression in children, atropine and pirenzepine, appear to block axial elongation in some of the animal models by pathways which do not involve accommodation, perhaps by acting directly on the retina or the sclera. By the 90s, the link found between education and myopia development in epidemiological studies was generally explained by the possible regulation of the rate of axial elongation by defocus during reading. In other words, the lag of accommodation present in humans while reading, places the image plane behind the retina, and it was suggested that this hyperopic defocus could induce axial elongation in humans, as it does in animal models. Bifocals and progressive addition lenses applied to myopic children showed some ability to slow myopic progression, consistent with this idea, although the effects were generally not strong, and it was not clear why some subjects developed myopia while others did not, if all children lagged to some extent while reading. Evidence on whether accommodative lag precedes or is a consequence of myopia is conflicting. More recent experimental data showed that children or adults with myopia had relative peripheral hyperopic defocus. Images in the peripheral retina fell behind the retinal plane in optically corrected myopic subjects, so it was postulated that the peripheral refractive error could drive the emmetropization mechanisms leading to changes in ocular growth patterns. More recent evidence suggests that the difference in eye shape may be a consequence, rather than a cause of myopia. In 2002 it was shown, in the chick model, that the stop signal produced by positive lenses was more robust than the grow signal of negative lenses. 1 hour of clear distance vision or exposure to positive lenses for short periods, i.e. images in front of the retina, cancelled the myopia produced in chicks by whole day of frosted occluders or negative lens wear. Experiments in monkeys with foveal ablation and lenses or occluders with central holes that allowed for clear central vision showed that defocus in the peripheral retina could significantly affect axial growth of the eye. This suggests that even if peripheral hyperopic defocus is not a cause of myopia, if images in the periphery are placed in front of the retina, myopic defocus, the stop signal could decrease axial elongation in human eyes. Spectacles and contact lenses with peripheral plus AD are being tested in humans, and there has been success in decreasing myopia progression in selected cases. More research is needed in this area before this technology is available. Thanks for watching.